In this lecture, we're going to take a look at prokaryotic cytoplasm construction, as well as looking at the membrane that surrounds the prokaryotic cell. So let's get started on that. The cell membrane is composed of material that is referred to as plasma membrane. So the cell membrane, by reminder, is the membrane that surrounds the entire cell. And plasma membrane is this living, dynamic material that the cell is capable of constantly modifying in order to adjust to changing conditions. It's built of a fluid lipid bilayer, and you hopefully recall from previous classes where you've looked at plasma membrane. It's built of phospholipids, so here is a phospholipid, and phospholipids are have a polar head that interacts with water, and then they have these non-polar tails that hang off of the polar head that don't interact with water and in fact find other phospholipids to interact with. And so that makes this oily layer here in the center of the plasma membrane. And that gives the cell membrane the capacity then to exclude stuff from coming in. So anything that has dissolves in lipid is able to pass very easily through the membrane, while anything that does not dissolve with lipids, so any polar molecules, will be repelled by the membrane. So it's studded, this lipid layer is studded with a mosaic of proteins, and so it's described as the fluid mosaic model for how membrane is built. So it's weak, relatively weak hydrophobic interactions that produce this cell membrane when the two tail sets of two different phospholipids find each other and interact with each other. So it's not a hugely structurally, it's not a strong membrane, I guess is what I'm saying. It's easy for osmotic pressure to rip apart the cell membrane. And so if there is no cell wall to help stabilize or structurally strengthen the properties of the membrane, then what we see is sterols like cholesterol molecules are found in the bacteria that don't have a cell wall. So as I mentioned, there are proteins embedded in the plasma membrane, and it's the proteins that produce the characteristics of selective permeability for the membrane, because these proteins can act as carriers, and when they act as carriers, they're able to transport materials from one side of the membrane to the other, and primarily what they're transporting is ions or materials that are too large to come in, or will not, they're polar materials that won't interact with that oily layer in the center of the membrane. So they might act as carriers, or they can form channels and pores that allow water, for instance, because water doesn't interact with that oily inner layer. So these pores and channels let water and other materials flow into and out of the cell relatively freely. So we see that those proteins produce this selective permeability, the ability of the membrane to let some stuff in while repelling other materials and preventing it from entering the cell. They can also be out here attached as identifier and receptor proteins. So these are proteins, identifier proteins, for instance, may be these kinds of glycoproteins where we have a protein embedded in the membrane and then a sugar that sticks off of it, where it acts as an identifier protein. And that it's those identifier proteins then that our white blood cells are able to recognize and then use as a means of removing the material from, or a bacterium from from our body fluids. And then we also see proteins that are attached here at the inner surface of the membrane, and they can be enzymes where they're working to help speed up the chemical reactions that are occurring inside of the cell. So those proteins that are found associated with plasma membrane, they've got a large number of functions. And so it's proteins then that give the membrane all of its function. These proteins regulate movement into and out of the bacterial cell. They act as enzymes to perform functions that in a eukaryotic cell are normally carried off by organelles. So they're busy, these proteins that are acting as enzymes, they're going to help to make new cell wall material, so making that peptidoglycan and other materials that are associated with the cell wall. They're involved in the production of new DNA, and they are involved in the chemical reactions that produce ATP. As well, if there's a flagella on the bacteria, then these proteins are going to be an attachment site for the flagellar proteins. So, as I was saying, lots and lots of functions that are associated with the membrane. So we're looking here at plasma membrane functions, and they are all given to them by, or they're primarily provided to the membrane. These functions are because of the proteins that are found associated with the membrane. All right, so that's everything that we've got to say about plasma membrane. So let's start looking at the material that's inside the cell, the cytoplasm. So cytoplasm is all the stuff that fills the cell, and it includes a material called cytosol. This is the gel. It's water plus a bunch of dissolved organic materials, especially dissolved proteins, that make the cytosol very gel-like. Now, the book uses the term cytoplasm to mean cytosol, and I disagree with the book. So this is one of those few places where when I say something, that I believe the book is wrong. And, I say one of those few. There's a couple in this chapter and the next, couple of points that I disagree with the book about. So cytosol is the semi-fluid gel, and then there are some organelles associated with the cytosol that then we together refer to the organelles, the other parts of the non-dissolved stuff that's inside the cytosol, to make up cytoplasm. So cytoplasm is everything. Cytosol is the fluid portion of the cytoplasm. Some of the organelles that we find in prokaryotic cells, and I want to step back for a second and say, we have said that prokaryotes don't have membranous organelles. So this is a difference here. There are no membrane-bounded organelles inside of prokaryotic cells. So we're not going to see Golgi apparatus or endoplasmic reticulum. But we do see that there are some organelles involved. And ribosomes are one of those organelles. You'll probably recall from previous classes, ribosomes are the site of protein synthesis. So that's where proteins get built. And it's found, these ribosomes are found in the cytosol, and sometimes they're grouped together in long chains where they're referred to as polyribosomes. And some of the antibiotics that we use to fight prokaryotic infections, they work because they bind to and then stop the function that's associated with these ribosomes. So they stop protein production in the cell. There's also a region where we find the membrane. It's called the nucleoid, or sometimes the nuclear region. So we have the nucleoid, which is just this specialized region of the cell where the chromosome is found, or if there are multiple chromosomes in the prokaryote, then there might be multiple there. But there's no nucleus or nuclear envelope surrounding the chromosome. and so we don't refer to that area as the nucleus. Instead, it's the nucleoid. So the chromosome is one or more either circular or straight strands of DNA. So the DNA is the chromosome, but associated with the chromosome there in the nucleoid can be bits of RNA and proteins. And so the bottom line is that there's a lot of variability among bacteria about what the chromosome looks like. Generally, we think of there being at least one circular strand of DNA in a chromosome, but in reality, there's lots of variability around this. And the job of the DNA then is to have the instructions for making proteins that the cell needs. And so the length of DNA and all of the nucleotides that are found in the sequence of the DNA is what tells the ribosomes how to make the proteins that it needs to make. And then there may also be structures called plasmids. Plasmids are accessory DNA strands, and they're also circular. And so these are just bits of DNA, maybe that the prokaryote, the bacteria, has shared with or received from another prokaryote, even in a process where they share genetic information with each other in the form of these little plasmids. So just little strands of DNA, or not strands, but loops of DNA. So a plasmid is like a little circle. So while there are no membranous organelles in a prokaryote, there are what are referred to as internal membrane systems. And these internal membrane systems are what they provide surface area for enzymes to do some of the more complex processes of the cell. So nitrogen fixation, that process where inorganic nitrogen is assimilated into a organic molecule, so that making it bioavailable then to the cell and to other organisms. That's nitrogen fixation. That's where we see the enzymes that do this very complex process of nitrogen fixation. And in those bacteria that do photosynthesis, all of the enzymes that run the photosynthesis reactions are found on these internal membrane systems. And then there are also structures that are referred to as inclusions. These are things that, again, not bounded by a membrane. There's no vesicle surrounding these inclusions, but they're just a dense, compacted, solid material that doesn't dissolve in the cytosol, and instead it's so densely compacted that it stays as a little granule. And so these granules might be glycogen. Recall that glycogen is a energy storage compound. It's a polymer of glucose, so it's just a bunch of glucose molecules that are all linked to each other and are there serving as storage function for all the glucose that the prokaryotic cell has either made or gathered. Polyphosphate is another one of these granules. Phosphorus is an important, or the phosphate ion, the PO4 3- ion. PO4 3-, that's the phosphate ion. So this is a polymer of phosphate. Phosphate's super important to a lot of cellular processes. And so storing up phosphate makes sense for prokaryotic cells. Excuse me there. And then there's sulfur. Sulfur is important to sulfur bacteria because these sulfur bacteria use sulfur in the processes by which they make ATP. So having sulfur around for sulfur bacteria especially is important, and so they will maintain a little storage granule of sulfur. Other inclusions include vesicles. So we do see vesicles inside of prokaryotes, and these do have a membrane around them. Inside the vesicle might be a gas. so they'll store gases in the vesicle, and that helps to make it, the more gas there is in it, then the more buoyant it is in water. The whole cell is in water, and so they can adjust the amount of gas. They can go to the surface of the water in order to photosynthesize during the day, and then drop down to lower regions of the water at night when they don't want to be so available maybe to a predator. Lipids may be stored in these vesicles, and so you probably may remember we looked at some chlorella cells, and they have a little lipid vesicle inside of the cell. And that's for storing energy and as a carbon source inside of the cell. And so lipids are a great way to store energy. And there may be iron compounds and other materials that are included in the vesicles. So granules have no membrane, vesicles do have membranes around them. And those are some of the inclusions that we see in prokaryotic cells. Okay, so the last thing that we might find in cytoplasm is a structure called an endospore. Endospores are a resting stage for the bacterium. So if the bacteria notices that, conditions are getting bad, or if there's damage to the cell in some way, then the endospore there is produced, and you can see endospores in these rods. So that's a specialized stain that's shown up now, this green, the endospore takes on this green coloration when you use endospore stains. This is another bacterium that's been sliced, and you can see the endospore here is this black area, and then it's surrounded by layers of membranous material and other things. We'll see in Chapter 6, we'll learn a lot more about how endospores are formed. But an endospore has two primary regions to it. There's the core, which is formed by a membrane of a layer of peptidoglycan that forms this outer core wall. And then inside is the nuclear region with a copy of the cell's DNA. And then it's surrounded by a cortex, which includes a spore coat, and in some, there's this outermost layer of exosporium. And so, we'll see more about this in Chapter 6. So as environmental conditions are degrading, then some bacteria will produce an endospore inside their cell, and when the bacteria die, then the prokaryote cell ruptures, and now the endospore is released into the environment, and it will persist there. As favorable conditions return, then the endospores can germinate, and that will produce a bacteria that will then go back to growing and reproducing. So I want to be really clear, this is not a reproductive strategy. If it were reproductive, then the cell would be making a whole bunch of endospores. This is just a mechanism by which the cell can outlast bad conditions and then come back to a vegetative state and continue to live, even though it dried out, or it was subjected to acid or to base or to radiation. And there are reports of 25 million year old still viable endospores. So they have a very, very, very long, I won't say life because they're in suspended animation almost, but they have a very, very long shelf life. How about that? Not all bacteria produce endospores, though, so this is something that we see in a relatively few number of bacteria. Okay, so there it is, everything you ever wanted to know about endoplasm. So what we see inside of a prokaryotic cell is the cytosol, ribosomes, a nucleoid region, and then these internal membrane systems, and then storage things like granules and some vesicles. And then finally, a hedge against hard times would be the endospore. Okay, as always, if you have any questions, do not hesitate to get a hold of me, and we can make sense of it.